Okay. We'll get a little bit on this. First of all, what's an allele? Right. It's a version of a gene. So instead of saying the version you got from mom or dad, you actually say we got an allele from mom or an allele from dad. This is kind of a review. Which of the patterns can't belong to the child of this couple? Let's see. So let's see. Which one? I'm looking at it. Okay, so let's see. This one, B? D. Yeah. Okay, so they got that from mom. No, and that from dad, so that's not D. That from mom.
C. Yeah, because C is a complete repeat of mother.
Yeah, right. So it's C, right? Mom and mom. Can't get two alleles from mom. Okay. Which cannot be the child of this couple?
Yeah, B. Because it's the same thing as the previous one.
B?
B.
D. Yeah.
Oh, okay. I didn't notice that one.
That's a double.
So it's B, C, D.
And dad and mom, see this one is okay because they both have the same allele, right? This one is mom. Hold it.
No, never mind. Because dad is also.
That's dad and that's mom, right? So yeah, so it's got to be. It's got to be mom and dad. This one, right? Okay. So what kind of cell is this? Eukaryote or bacteria or archaea? Huh? Eukaryote, right. Because it's got a nucleus and all these other organelles. So who are these guys? Right. Oh, by the way, they got a Nobel Prize for that. So what's the complementary for this? Which one for this? G. C. C.
A. T.
Right? Okay. DNA polymerase. The double-stranded molecule unwinds an enzyme called DNA polymerase, then adds complementary nucleotides. So, in other words, each of these strands is a parent strand, and then a new strand, when the cell divides, complements the base pairs. Right? So, let's say this one will have to be this one, the complement, right? So what it does is it actually starts adding on, let's say the parent strand is, okay, there we go. So here's the parental strands, right, along here. And then on the inside is the complementary base. Okay. What procedure will a forensic scientist do? We already know that. PCR, amplify it. Okay. You can read this yourself, but you can get a genetic profile with a number of repeats. Remember how this goes? So larger fragments move more slowly. Smaller fragments move more quickly because they're lighter. Okay, so here's a genetic code. Like I said, there's 20 amino acids. If you look at these letters, like TAT, that codes for tyrosine, which is an amino acid. You need 3 letters to make an amino acid, or a code for a certain amino acid. It's always, a codon is always 3 letters. Now, if you're using the transcript, which is made of RNA, then you actually, instead of using a T, you use a U. It's called uracil, but that's besides the point. Okay, so there's 20 amino acids, 3 base pairs long, code for an amino acid. Some of these are actually redundant, and then some actually code for what are called stop codons. Okay. That's good. We'll, I think next week is a test, so we're going to have to pick up where
That's not right.
Next week?
I think it is, right?
The week after?
It says the 23rd, but Friday is the 21st.
What?
It says the 23rd, but Friday is the 21st.
Okay.
I'm assuming.
Yeah. That seems so soon.
Yeah, because we had a test last week.
Yeah. I don't know. Would you like, maybe we should like, would it be okay if we slipped it down to the next Friday?
Sure.
Yeah, I'll send an email and I'll see how people feel about it.
I bet half the people don't even know.
Take your sweet time, I got nothing to do.